Hi, so right off the bat, I want to apologize for not being as creative with a song or anything fancy like that. So instead, I'm just going to talk myself through the alternation of generations that all plants go through. So for the practical, you and I talked about like all the different structures, all the different characteristics of plants that we're going to be going over, but the one constant is how they reproduce, which is the alternation of generations. So it all starts with a diploid sporotype. I thought it was very convenient, this little chart here. This chart really goes through and gives a good demonstration of this process. As you can see, all the diploid parts of this process are in green, and all the gameotype portions are in blue. That line across really demonstrates which parts are each, and helps me to kind of keep track of this process and when it starts with the gameotype, where it ends with sporotype. Like I said, or like you said today in lab on February 16th in lab 5, this process will be critical to know to be successful in the practical. So I'm just going to go through it real quick, and I feel pretty confident with it, but practice makes perfect. So like I was saying, the process starts with a diploid sporophyte, and then undergoes a process called meiosis, which is when something splits into two different cells. Kind of had to go back and review some biology 1 concepts just to make sure I could differentiate between meiosis and mitosis. Mitosis meaning that two daughter cells are identical after the split, and meiosis is two different. So once you get that kind of mastered, you look for the keyword meiosis, and you know that you're going to be going from diploid to haploid. So after the sporophyte undergoes meiosis, it will go and split into two different spores, which are going to be haploid gameotypes, which are Archegonia and Antheridia. Archegonia being the female, which is the egg, or at least Archegonia will undergo mitosis, and the Antheridia, I think. Yeah. Both go through mitosis to produce an egg and a sperm. This is where I think a lot of people are going to get messed up, because fertilization is a key word here. That means that the two haploids are going to come together to form a diploid zygote. The easiest way for me to remember it is if you have two halves and they come together, you have one. So that would be the diploid, even though that's two. But for me, it makes sense. I guess that's what's important. So after you have your egg and your sperm fertilized and they form into a diploid zygote, they will then go through mitosis once again, meaning two daughter cells that are completely identical. And those that next part of the stage is a diploid embryo, which will then go through mitosis again and start back over with diploid sporophyte. You mentioned heterospory, but heterospory, which is, the mega or microspores. Mega is the female, micro being the male, but I do believe at the time I made this video, we hadn't exactly gone over that. I believe that's for lab 6. So I know I probably messed up a lot of the terms. I'm sure I missed a few steps, but overall I feel really confident in this process, and it should help me study the rest of the material. And like you said in class, everything might be different characteristic wise, but at least this one process, if you can label that, you'll be successful in certain parts of the practical. I hope you learned something new. I sure did. Thank you for the extra credit opportunity.